DNA is made up of sugars, phosphates, and bases. One group of each of these three components forms a nucleotide. DNA strands are comprised of many nucleotides joined together. DNA is a two-stranded molecule, and the two parallel strands are joined by hydrogen bonds between the complementary bases. There are four bases: adenine, thymine, guanine, and cytosine. Adenine is only complementary to thymine because they are joined by two hydrogen bonds, and guanine is only complementary to cytosine because they are joined by three hydrogen bonds. DNA is the code for the synthesis of proteins known as polypeptides, which are the building blocks for all cells and organisms. Small sections of DNA, called genes, code for specific proteins with specific functions. The first step of DNA's transcription takes place in the nucleus of the cell. RNA polymerase splits the DNA at the promoter area of the gene and uses one of the exposed strands as a template for the formation of the complementary mRNA strand. The RNA polymerase enzyme builds the new mRNA strand by pulling free-floating RNA nucleotides from the nucleus and adding them to the 3' end of the strand. This new mRNA strand is complementary to the original DNA template strand. However, the DNA-based thymine is replaced by the RNA-based uracil. Once the RNA polymerase reaches the termination sequence point of a gene on the DNA strand, it detaches from the DNA strand. Splicing to remove non-coding sequences on the mRNA strand occurs and the strand leaves the nucleus. Once it enters the cytoplasm, it moves towards a ribosome to begin translation. This is where the polypeptide synthesis takes place. The mRNA strand enters the ribosome and is read in groups of 3 nucleotides known as codons. Each codon on an mRNA strand dictates the specific sequence of amino acids in a polypeptide chain. Transfer RNA molecules containing complementary bases arranged in groups of 3, called anticodons, match with the mRNA codons. Each tRNA molecule is coded to carry a specific amino acid. As the tRNA anticodon matches to its complementary mRNA codon, its amino acid is released, and the amino acid forms a peptide bond with the previously released amino acid. This process repeats until a polypeptide chain of many amino acids and peptide bonds is formed. The processes of DNA replication and polypeptide synthesis occur in the S phase of the cell cycle.